Meiosis presentation by Abigale, Kate, and Ty. A diploid is a cell consisting of two sets of chromosomes, one from the mother and another from the father. A haploid is a cell consisting of only one set of chromosomes. Homologous chromosomes are just chromosome pairs, one from each parent, and they're similar in length, gene position, and centromere location. The position of the genes on each homologous chromosome is the same. However, genes may contain different alleles. Bivalent is a pair of homologous chromosomes. Synapsis is the fusion of chromosome pairs at the start of meiosis. Crossing over is the exchange of genes between homologous chromosomes, which results in a mixture of parental characteristics and offspring. Chiasmata is the point in which two homologous non-sister chromatids exchange genetic material. Random orientation occurs in metaphase 1, and the orientation of molecules is random, meaning that there is an equal probability that the daughter cells have either the maternal or paternal chromosome.
Interphase 1, or just interphase because there's only one phase of interphase, is the largest part of the cell cycle. It consists of a time where they perform normal cell activities, like metabolic reactions. At this point, the chromosomes are still coiled up bits, and they're beginning to coil, and the nucleus is still around. In prophase 1, the nucleus begins to disappear, and the spindle fibers start coming out of the centrioles that move to the poles. The replicated DNA from prophase 1 begins to find its bivalent and begins to form a bivalent and crosses over. In metaphase 1, the bivalents line up in the middle of the cell, and the centrioles send their spindle fibers out to reach towards the middle section. In anaphase 1, the spindle fibers attach to the centromeres of the chromosomes and pull apart the bivalents to the polar ends of the cell. In telophase 1, the centrioles begin to break down as well as spindle fibers, and the chromosomes move to the poles and pull apart, creating a cleavage furrow. In prophase 2, the two cells
Condense. Again, nuclear membranes dissolve and centrioles duplicate in spindle fibrous form.
In metaphase 2, the chromosomes line up in the center of the cell, and the spindle fibers attach to the centromeres from the centrioles.
In anaphase 2, sister chromatids are pulled apart and brought to the poles. They are now identical chromosomes.
In telophase 2, both cells begin to come apart again, creating a cleavage furrow as they split.
In cytokinesis, the cell membrane pinches and separates, forming 4 gametes.